So, dear colleagues, today is a day for last seminar this year, and we have today a guest from California, San Francisco, Professor Brian Shoichet, who received his bachelor in chemistry from Massachusetts Institute of Technology and PhD in medicinal chemistry from University of California, San Francisco, for work with Jack Kuntz on molecular docking. Shoichet did postdoctoral training at the Institute for Molecular Biology in Eugene, Oregon, with Brian Matthews, studying how enzyme activity traded off with enzyme stability. He started his own research lab at Northwestern University in Chicago in the pharmacology department, where he received tenure in 2002. In 2003, he was recruited back to University of California, San Francisco, where he is now a professor of pharmaceutical chemistry. His studies to early techniques in ligand discovery with particular focuses on large library molecular docking, chemoinformatic inference for target identification, and the role of colloidal aggregation as confounds in early discovery. According to Scopus, he has about 280 publications with circa 50,000 citation. Each index is 107. And dear colleagues, I ask to keep your microphone switched off. And dear Brian, so microphone is yours.
Thank you. Can you hear me?
Yes.
Terrific. Well, I'm very honored to be here at Kharkiv National University. Thanks to Valentin for the very kind offer. Thank you all for staying late in your day, early in mine, to hear about the research we're doing. I thought I'd begin by giving you an overview of the topics that I'll touch on today, and I'll come back to this so you know where we are in the talk as time goes on. The first I'll introduce ultra-large library docking, um, the the expansion of chemical space of really accessible molecules that has allowed it. And this is really an innovation of Ukrainian chemistry, for which we're very grateful. I'll talk about one application of the method to the calcium sensing receptor, where we sought to get novel molecules with potent activity and different novel pharmacology than what we've had before to treat a disease. I'll talk about new work, unpublished, on how good we are at optimizing molecules that we find for affinity and for pharmacological properties. As you'll see, we're very bad, but I hope it'll teach something interesting. And then finally, I'll come back to the early theme, which is the massive expansion of accessible chemical space that we have experienced, that the field has experienced, and whether it's, when we quantitatively assess it, whether it has improved our outcomes. And what I'll argue is that as the library has grown larger and larger, our results have improved, and we try and convince you of that quantitatively, and are not plateauing. So it seems like the larger libraries lead to better results, and there's every motivation to keep on expanding them, except we don't know how. That'll be a technical challenge for the end. I should tell you about my conflicts. These are they. This is something UCSF asks me to do. Okay, so this is large library docking as it was about 5 years ago. So we start off with a library of accessible molecules, molecules that somebody somewhere would sell us, which 5 years ago was about 3 million available molecules. It's important that we be able to get the molecules quickly and easily because docking has many failures. And so one way to mitigate that is to make the cost of failure cheap. And if you can get molecules relatively quickly and easily, molecules that the docking predicts quickly and easily, then it's not a tragedy if the first one doesn't work because you can go on to the second one and the third one and the 100th one. Each molecule in the library is docked into the binding site of your target in hundreds of, well, I should say thousands of configurations, so different physical poses, rigid body poses of the molecule. And for every configuration, hundreds of conformations, internal conformations of the small molecule are sampled. So overall, about 1 million different orientations and conformations are sampled for each molecule in the library, and each is scored using a physical chemistry-based scoring function involving van der Waals interactions, for instance, from the amber potential, Poisson-Boltzmann electrostatics these days from QNIFT, from Kim Sharp, and several other terms. We desolvate the molecule, so we calculate the cost of bringing the molecule out of solution and subtract that from the electrostatic energy and so forth. And the terms themselves are all terms that are used, but they're leading edge terms in the field. But we do leave out some important terms, and even more important is we don't relax the system. We don't allow the protein to change conformation or to minimize its energy when the ligand is fit into the binding site. We do sample multiple conformations for the ligand, and we don't relax the electrostatics because the ligand binds, the boundary conditions change, and we don't consider that. So many approximations are made. Nevertheless, the method, which is, I think, unique among the major theoretical methods, tests its predictions experimentally. That's always been central to the whole approach, and we continue to do that. The big change, I would say, in molecular docking, which has incrementally been improving for 30 years, the big recent change, the disruptive change, is that our libraries have really popped. We were docking 3 million 5 years ago. We're docking 6 billion today. And the number we could be docking is over 75 billion. And the reason we're not docking 75 billion is the libraries have expanded faster than we can keep up with physical modeling. Why have the libraries expanded so much? And this is the contribution of Yuri Moroz and his colleagues at Enamine, which is the introduction of what my colleague Adam Renslow calls tangible molecules, which are molecules that don't exist but could rapidly exist if you wanted them to. And what Enamine pioneered is this creation of interesting building blocks that they have on site in Kyiv in 10-gram quantities. The building blocks are to a chemist interesting looking. They are highly stereogenic, many fused and spiral rings, and interesting functionalities. And they can, over 30 years, enamine has developed reactions, applied reactions that can put subsets of them together reliably in 2 and 3 component, sometimes 4 component reactions. And when you do the combinatorics, this leads to about, to over 75 billion possible products. And because the chemistry of the building blocks are interesting. The chemotypes explored in the final products are also interesting for us. And so our challenge as a molecular docking lab has been to keep up with this expansion, which continues to outstrip our abilities. But it's for us, it's a good problem to have because the molecules are very interesting. Um, Anecdotally, so by example of particular stories, it seems like in our experience and the experience of others in the field, that library growth has led to better results, better docking results, and better pharmacology for the compounds we're trying to discover. So, this is just what this slide is trying to do is convince you to hear the rest of the talk. So it's sort of a trophy case of how of where docking has worked. So an early success was against the melatonin receptor, which is one of the main receptors controlling circadian rhythm response to light and darkness. This was done with Brian Roth and Margarita Dobokovic, and right out of the docking against this G protein coupled receptor, we got low nanomolar to high picomolar uh, uh, antagonists and agonists of the melatonin receptor. And in work with Andy Cruz's lab, we applied docking to this dark horse of pharmacology, I would say, the sigma 2 receptor, and likewise got low nanomolar actives directly out of the docking. And these were applied after some optimization to test the role of the sigma 2 receptor in analgesia with Alan Basbaum's lab. They were analgesic. And here and actually in every other target I'm going to show you, the docking predictions were tested crystallographically, and you can't really see it very well. I apologize, but I'm showing the superposition of a docking prediction on a crystallographic result solved in the Cruz lab by Asaf Alon. And the reason you can't see two molecules there is because they superpose basically exactly. So that's an example of docking being right for the right reasons. Same thing with the alpha 2A adrenergic receptor, also for analgesia. These molecules were ultimately potent in a mouse model for pain with Alan Basbaum, and once again with Yang Du's lab in Hong Kong, we showed that we solved the structure and the docking was right for the right reasons. And I won't belabor this too much, but in multiple other targets, many against G protein coupled receptors, many in the central nervous system because that's been our focus, but also against COVID-19, against some ion channels for cystic fibrosis. The method has been applied and got relatively potent molecules against diverse targets. So that says that the method with what we call a good binding site, a well-formed binding site suitable for a small molecule, and with some ligand precedence so we can do control calculations, the method can work. What I'm not showing you is the examples of where it's failed, because we certainly have used it and failed or struggled. Like for instance, I'm not showing we have a case against main protease of COVID-19. That work's also published and soon to come out. And eventually we made it work. We eventually got very potent molecules working with Adam's lab, but it was slow going. It was a real struggle. Hit rates are low and the compounds were not potent initially. And that reflects a difficult binding site. I should also say none of this would have been possible without a lot of collaborators around the country. Brian Roth has been very central to our works for 20 years, but also my friend and colleague Alan Basbaum at UCSF, Ashish Manglik, and many others that I don't have time to really properly acknowledge. I would like to take you through one example in a little bit more detail. And this is a story that just came out against the calcium sensing receptor. Sensing receptor is a homeostatic receptor, one of many homeostatic receptors in the body. It's involved in uh, helping to control serum calcium levels and, by extension, parathyroid hormone levels. And the parathyroid glands are these little dots on the thyroid gland here. I hope you can see them. And they release parathyroid hormone, which has many roles in the body as sort of what we call pleiotropic effects, but among them, it controls vitamin D release and controls calcium levels through absorption or reabsorption back into the circulation of bone calcium. And it also has a role in the excretion of calcium through the kidney. So parathyroid, one of its central roles is calcium control. And this calcium sensing receptor, when it's activated by rising, so parathyroid hormone acts to release calcium, and as calcium is released, it activates the calcium sensing receptor, which puts a brake on the secretion of further parathyroid hormone and puts a brake on the parathyroid's hormone's effect on the kidney, leading to more excretion. And so the result is that serum calcium is very tightly controlled. The homeostasis in your body of calcium is very tight. It only ranges between 2.2 and 2.6 millimolar through the action of this system. Um, in some diseases, nevertheless, calcium concentration is dysregulated. In cancers of the parathyroid gland and several other cancers, parathyroid release is dysregulated. And also for patients on kidney dialysis, calcium homeostasis is disrupted. And so drugs like cinacalcet here have been released. It's as well as Evocalcet, only in Japan for Evocalcet. And these drugs activate the calcium sensing receptor to help to restore homeostasis. And cinacalcet has just gone off patent, but while it was on patent, it was selling close to $2 billion a year. So it's a big market. But the drugs are imperfect. They have problems. One of their major problems is um, they easily can lead to hypocalcemia, so they overshoot their effect, and so the calcium concentration drops too low, and that leads to heart arrhythmias and eventually heart attacks. So it's a serious dose limiting side effect of the drug. Um, we know a lot about how the drugs work at a molecular level via their interaction with the receptor. Those structures were solved by Yorgos Skiniotis and his colleagues at atomic resolution in papers that came out over the last 4 years. And the the calcium sensing receptor is an obligate dimer. It acts as a dimer of monomers. And in the dimer, there's thought to be an active and an inactive monomer. I'll refer to the active monomer as the B monomer. Here, it couples to G proteins. Calcium sensing receptor is a G protein coupled receptors. They're called a GPCR. They're called GPCRs because when they get activated, they activate a downstream enzyme called a G protein, and that protein. Um, but with this obligate dimer, only one G protein. It's a dimer, but only one G protein binds. So there's an active and an inactive monomer. And in the active monomer, cinacalcet, shown here in orange, adopts a bent conformation in the binding site. It folds over itself. And in the inactive conformations, it's extended. So What Fengyu Liu, a very talented postdoc in the lab, did was launch a docking campaign against the calcium sensing receptor. She actually launched two campaigns, one with our old library, the 3 million molecule library, and one with the 1.2 billion molecule library. I'll focus on the large library here. And as ever, each molecule was docked in about 1 million configurations in the binding site and scored for fit. And from the best scoring molecules of the 1.2 billion, she prioritized probably about 90 for synthesis, and 74 were successfully synthesized by our colleagues at Enamine. And of those, and they were tested in Yorgo's lab by Cheng Guo Wu, and of the 74, 24 were active. So 37% of the molecules we predicted to be active were active, with EC50s with activities ranging from 300 nanomolar to 30 micromolar. And they were selected for two things. They were selected for being chemically different, so not to resemble topologically cinacalcet or evocalcet, the known drugs, and to be diverse among themselves. So they were sort of doubly diverse. Don't resemble the knowns and don't resemble each other. So we would try and span a large range of new chemical space. So that was successful in itself by docking standards, but the molecules were not super potent and needed to be optimized for pharmacology, and so we Fengyu did that against several of the more interesting chemotypes. So, for instance, this molecule 5250 was optimized to this molecule 2021, largely by stereochemical purification and the addition of a single methyl group, but that improved activity about 200-fold. The same thing with this molecule, 5670, began much weaker at 14 micromolar, but we were able to optimize it to, also by about 100-fold. And also, I should also emphasize that we were conscious of the hydrophobicity of these molecules. One of the problems with cinacalcet is it's a very greasy molecule. The calculated octanol to water partition coefficient, the C log P, is 5.1. So that's like 100,000-fold preference of water for octanol, so very greasy. And we wanted to keep the new molecules more polar because we thought they'd behave better because of that. And we were largely successful in doing that. And so this molecule starts at 30, so pretty weak, but here we were able to optimize it about 1,000-fold. And there's two arrows shown here in most places because we often multiple rounds of analogs were synthesized, often 30 analogs at a time, 3 rounds. So this was a bit of a slog, even though it was successful ultimately, and I'll come back to that, what that means. Two of these molecules we took forward. This is 2460, which I think is ultimately the 100 nanomolar molecule. This is showing that Cheng Guo solved the structure by cryo-electron microscopy. This is the dock pose is shown in blue, carbons in blue in both cases, and the cryo-EM result that Cheng Guo subsequently got is shown in purple. And you can see in the inactive conformation, the docking was not perfect, but basically right for the right reasons. Same in the active conformation, all the key groups are making the proper key interactions, but there are some interesting changes. For instance, this aromatic group swings down by about 45 degrees, and correspondingly, groups on this recognition helix of the protein swing into the binding site. those were changes that we did not anticipate in the docking. Similarly, for this molecule, this 54149 molecule, also broadly correct. Docking prediction superposes well on the cryo-EM result, but here also important changes, especially in this aromatic ring, this benzothiazole aromatic ring is really flipped by now 90 degrees, and there's even more changes in this activation helix of the receptor. So broadly right for the right reasons, not perfect in every detail. Okay, but I promised you some pharmacology. So we were interested in finding novel chemotypes for two reasons. One is we thought we could improve on potency, and the second one is we were hoping that the novelty of the chemistry would improve the hypocalcinemia that is the major dose-limiting side effects of these drugs. And this is work done by Wenhan Chang, shown here, collaborator at UCSF, and in ex vivo studies on isolated parathyroid glands, he showed that compared to cinacalcet in green, this new molecule, this 30 nanomolar novel allosteric modulator activator of the calcium sensing receptor, was more potent at suppressing parathyroid hormone release. So this is parathyroid hormone release from the isolated glands on the y-axis, and this is concentration of the drugs. And here's cinacalcet in green. You can see it's actually relatively weak. And here's the new molecule in orange, much more potent. And you can do this is holding calcium constant and varying the drug, but you can do it the other way around. You can hold the drug constant at 50 nanomolar, that's what the value we chose, and vary the calcium concentration, because the drug is a positive allosteric. It binds in a different site. It's an allosteric modulator of calcium. And here, the difference is more obvious. This is green is cinacalcet, again, the blockbuster drug. And here's the new molecule. And the y-axis is, once again, parathyroid hormone secretion. So that's an ex vivo study. You can also do these studies in vivo. To do that, you need to know the pharmacokinetics. This is showing the exposure of the drugs, and I won't go into it because I'm worried about time. But you can see that the new molecule gets exposed systemically in the mouse when dosed by intraperitoneal injection. This is now an in vivo result on live mice where we're measuring circulating parathyroid hormone in the blood, and we're comparing the new molecule against cinacalcet. And once again, so this is a 10 milligrams per kilogram dose of both the new molecule and cinacalcet in green. But as you can see, as you drop the dose from 10 mg per kilogram to 3 to 1, the cinacalcet effect quickly goes away. But the new molecule is more potent and continues to have effects down to 0.1 milligrams per kilogram. So more potent also in vivo. And then the most interesting result is that the new molecules do not suppress calcium homeostasis. They do not lead to this dose-limiting side effect, which is hypocalcinemia of the established drug. So here's cinacalcet. Now this is time on the x-axis and calcium concentration on the y-axis. And after a 3 mg per kg dose of cinacalcet, so that's a dose where cinacalcet is actually already losing its effect, you see this profound hypocalcinemia and basically nothing for the new agent. And so this is an example of how, starting with the structure, screening a large library, you can get novel chemotypes, optimize them for potency, and the novel chemotypes lead to new pharmacology. And that's something that it's hard to always logically explain why it happens because new chemistry doesn't have to lead, binding in the same binding site doesn't have to lead to new pharmacology, but it's frequently something we've observed. Okay, so I'd like to talk. I've mentioned our challenges with optimization. We frequently, in favorable circumstances, the docking works, and we get new chemotypes, and then our challenge is to optimize them. And that's something with which we've really struggled. So if I, go back to this trophy cases of where docking has worked, and I always show two arrows, and that means we almost always have to optimize them. Um, and I look back at how many analogs we've explored. Um, it's over 700. And only 5% of them improve. So only like 35 or so improve activity in a single step by more than tenfold. And, we're supposed to be good at this, structure-based design, look at how the ligand binds to the pocket, say, oh, we can put another phenyl group there or a chlorine there, and it should improve activity. And sometimes it does, but it's rarely that it's a really big change, tenfold or better. And I thought that was disappointing. Um, but then the thing that was even more disappointing is I didn't know if it was good or bad or mediocre. I mean, compared to what? And in medicinal chemistry, unlike our biology colleagues, we don't have like random background controls. We don't know what the random expectation is for a perturbation to a small molecule to affect affinity or any other property. And so I don't know if 5% is good or bad, or right. And so we thought we'd explore that. So what we have been trying to do is put together a null model for medicinal chemistry. What is the expectation um, of an affinity change, but you'll see there's other things too, for a small perturbation to a molecule? What change should you expect at random? And so so far we're up to about 278 minimal perturbations across 18 starting points, 18 different scaffolds. They don't resemble each other, or one or two do, but mostly they're different scaffolds from six different targets, four integral membrane proteins and two soluble enzymes. And what we've done is we've started, like, for instance, this is an actual starting ligand. It's a ligand for the alpha 2A adrenergic receptor, and we've just gone around and systematically and comprehensively replaced, like we'll take a ring carbon and replace it with a nitrogen. Or we'll take a hydrogen off the aromatic ring and replace it with a methyl or a fluoro or a chloro or a hydroxyl. And we'll do that without thinking, just systematically, comprehensively make those changes only one at a time. No more than one change per molecule, wherever the molecules can be made, relatively inexpensively. Um, uh, and so here's an example of, for this molecule, the results we got. And so, like in some places here, a single methyl would improve activity 18-fold. And the nitrogen change, this carbon to a nitrogen, would reduce it like 300-fold. And then in other places, we only, like here, the methyl change would only change it twofold. So only modest changes. And so we just looked at all of them systematically and comprehensively and asked, how often with these, what we consider small, random perturbations, only one at a time, do we improve affinity more than tenfold, right? When we're trying in my lab over the last 10 years or so, 5% of the time we succeed. So here's the results. So for alpha 2A, which is GPCR and responds to norepinephrine, an adrenaline receptor. So we had four different starting compounds. Uh, we had made for the first one, for compound one, 32 analogs were made by single perturbations, and three of them improved more than tenfold. And for compound three, 17 analogs were made, and none improved more than tenfold, and so forth. And then for cannabinoid receptor, a different GPCR, same thing. For the mu opioid receptor, you know, same story, and so forth. And over up till now, we hope this number is going to go up a little bit, but we've made 278 of these analogs, and I would say about 36 improve tenfold or better. One or two of them like are eightfold, but I count them too. That means, so in this random, comprehensive, systematic, small changes, 13% improve tenfold. So that's far better than what we've experienced when we're trying. And B, we think it suggests actually a method to explore early on your molecules with PharmChem, a very sort of no thinking method. But most importantly, it's for the first time it goes towards establishing a random expectation background for what you should expect. If you think you're good at medicinal chemistry, we would say you should be able to beat random, which is looks like about 13%. So it turns out, of course, that uh, affinity is not the only thing that's important for progressing medicinal chemistry. The how it behaves in the body, the pharmacokinetics, the PK, is also important. And so we looked at the in vitro pharmacokinetics, the harbingers of how these guys are going to behave in the body as well. So we looked at this. This is work with enamine, I should say. We looked at the solubility of the molecules, how much they were bound to plasma protein. So it's the free fraction that's important in pharmacology. Their stability in the plasma, their stability when exposed to liver microsomes, how they were permeable through membranes, and their ability to inhibit this tox target hERG. And without going through all the data, what I can tell you is there was no correlation between affinity and these other properties. In fact, they were typically anti-correlated. So an improvement in affinity would lead to a molecule that was less stable for microsome stability, or was less permeable through cells. So it seemed like these very important parameters trade off against each other, and it's hard to get a molecule that's perfect by all criteria. And so that's an illustration of why medicinal chemistry is difficult. This says the same thing in a different way. Here we're looking at the individual changes, the single atom perturbations that led to more than tenfold improvement. So the easiest way to get tenfold was with adding a methyl group here, and whereas aromatic carbon to nitrogen rarely improved affinity. But for the pharmacokinetic properties, so for instance, microsome stability, methyl groups sort of rarely improved my stability against the liver microsomes, but nitrogens often did. So that's and same thing for fraction unbound in the plasma. Nitrogens often would improve things, methyls rarely did, but it's the methyls that improved affinity more often. So that's another example of how these things trade off against each other. Okay, in the final few minutes, thank you so much for your patience. I'll talk about we've invested a huge amount in expanding our libraries from docking. It's a huge computational effort, a huge investment in infrastructure. Has it paid off? And what I'll try and convince you is that it has and will continue to. So we've explored this two different ways. this question of, do we get better results from our libraries growing? One is purely computational. And the question is as we get to larger and larger libraries, do our docking results improve? Does the fit of our molecules as measured by docking score get better? So this is work done by J.K. Liu, working with John Irwin. And what he's doing here is he's on the x-axis growing the library from initially 100,000 molecules to here just over 100 million, and looking at the top scoring molecules. And what you can see is the top scoring 5,000 molecules, which are different for every library, their scores steadily and monotonically improve as the library gets bigger and bigger. And this is against the dopamine receptor, and it looks what we call log linear. So for every tenfold improvement of the library, we get you know a, a constant improvement in the docking score, which is the fit of the molecules in the binding site. And if we go from here 138 million to over 300 million, we see that improvement continues. And we also, JK also did it against the serotonin receptor with a larger library and saw the same thing. And similar story for the sigma-2 receptor. As the libraries get bigger and bigger, the very top scoring 5,000 shown here, their scores steadily improve. You can see that the very top molecule, which is shown in blue, the top scoring molecule at any particular library level, improves more than the bulk of them do. And that's probably true not only for the top scoring molecule, but the top 10 or 20. And this is an example of where we're starting to get false positives from the docking. These are molecules that they diverge, they get much better scores than we'd expect. And the reason they do is because they're starting to cheat the scoring function. And that turns out to be a problem, but it's, I think, a problem we can address. I'll come back to that. Okay, so I told you we've done it two ways. This is the second way, which is experimentally. So if we compare docking a small library against docking a large library, prospectively, when we're testing new molecules, do our hit rates improve? So remember, I told you that Feng Yu tested two libraries against CASR. One, a library of 2.7 million molecules, so basically our old library from 5 years ago, and then a 1.2 billion make-on-demand library. And she compared the hit rates of activity. So I told you before that 74 molecules were synthesized and tested. And then from the small library, she prioritized 48 for testing. And here's the docking results. You can see that from the scores on the x-axis, and more negative is better, that are left shifted. So the 1.2 billion campaign, by and large, achieves better scores. And this is the number of molecules from the library achieving a particular score. So that this distribution is left shifted, and that's what we would have expected from JK's work, which I showed you on the last slide. And then when we go to actually physically testing the molecules, so this is for all actives here on the left. And the y-axis is the hit rate, and you can see that from the small library, the hit rate, just so the percentage that were active, is 13%. From the large library, it was like 37%, so almost 3 times better. And also, if you look at the range of actives, so these from the small library, I'm showing you all actives, but none of them were better than 10 micromolar as positive allosteric modulators. Whereas from the larger library, we got molecules that were in the 1 to 10 micromolar range, and also the 100 nanomolar to 1 micromolar range. So not only a better hit rate, but also much more potent molecules from the large library. So Feng Yu also wanted to do this on beta-lactamase, which is sort of a model system in our lab, and do it at a bigger scale. So JK had, in the first application of the large libraries, had screened about 100 million molecules against beta-lactamase and had tested 44. And Feng Yu now with a larger library um, uh, tested about just over 1,500, so massive expansion. Um, Same thing as the libraries get bigger, as this 2.1 million billion, sorry, molecule library gets bigger and bigger, the scores improve. But we start to see these molecules that score much better than everything else in the distribution. There's not many of them. This is like 100 out of 2 billion. So they're not many, but the these are almost certainly, well, we've shown that they're artifacts in subsequent studies. And there's not many of them, but they rise to the very top of the scoring list. And so without demonstrating that they were artifacts, Feng Yu wisely chose not to test any of the molecules in this range. Um, and and here's the results that she got when she compares docking 2 billion to 100 million. So for these are inhibitors of this enzyme, beta-lactamase. When you look for molecules that are better in between 100 nanomolar and 1 micromolar, there were several. So hit rate was like 2% or something from the large library screen, but there weren't any from the smaller library. When you go from 1 micromolar to 10 micromolar, now you start to get molecules from the smaller library, but at a much lower rate than from the large library. And, same thing as you go less and less stringency on what you define as a hit. So many more molecules, a much higher hit rate of molecules was found from the large library screen than the small library screen. And we reached higher affinities. So that's also consistent. So once again, experimentally consistent with bigger is better. Um, and we also got some a surprising result from this study. So this is what I'm now showing you is how hit rate changes with docking score. And so docking score better is to the left, more negative, and higher hit rate is obviously better. And you can see that whether your cutoff is like, oh, they need to be better than 150 micromolar, which is pretty weak, or a little bit more stringent, better than 50 micromolar inhibitors, or better than 15 micromolar inhibitors. You see this same pattern where the hit rate at low scores, at bad scores, the hit rate is nothing. And then eventually it starts to climb. And it climbs steadily and monotonically and never stops. It doesn't plateau. And this is very interesting for us because well, for one thing, it suggests that better scores lead to better hit rates, which is great. Not certain because of the approximations in docking, but very exciting. And the fact that it doesn't stop, it doesn't plateau is something new. What we'd previously seen in earlier studies where we test hundreds of molecules is that it did plateau. And here it doesn't. And I think the reason it doesn't is that we don't test from the very top scoring 100 or so molecules, which we believe are artifacts. And so we don't get confused by those, whereas earlier screens we did. And then the final thing that was very surprising is that if you notice the blue curve, which are the weakest molecules, starts to go up at, let's say 74 or something docking score units. And the red guy, the more stringent molecules only start going up around a little bit over 80. And the most stringent, the best performing molecules, they only start to appear at the best scores. And what that teaches is that there is a weak but perhaps meaningful categorical correlation between docking score and compound affinity. And we wouldn't say that at all, except this when we go back and analyze earlier results, also removing these artifacts, we see something similar. So this is an earlier campaign that published a few years ago, where we've now removed in retrospect, we've removed the artifacts. And similarly, molecules bind better to sigma 2, so the blue line is, better than 400 nanomolar the green line is better than 40 nanomolar, but you see the same trend of the weaker molecules come in at worse scores than the more potent molecules. So maybe there actually is a correlation between docking score and affinity, something we've never believed before and still don't fully believe. Okay, gone on longer than I meant to. I think I'll conclude by, um, leaving you with what I think the most important message is, which is accessible chemical space is exploding. This is largely the work of uh, your colleagues at Enamine, our longtime collaborators there, and computationally our lab and a few other labs. So it's very exciting. We expect that pretty soon the libraries will grow from now hundreds, close to 100 billion to into the trillions. In terms of what we're docking, right now we're at, 6 billion or something. I think, and we're doing everything brute force. Every, 5 billion molecules, every single one is docked and scored and so forth. But I think our ability to do that will fail pretty soon. The raw capacity to dock molecules brute force. It's been great, but I don't think we're going to be able to do that through many more expansions of the library. Um, and meanwhile, so around 20 billion we fail. The library is already a lot bigger than that. And like I say, in a few years, I very much expect it to be into the trillions. And so how to access those new space, what I've tried to convince you in this last section is that as the libraries get better, get bigger, our results improve. And so we should expand into the trillions. But how exactly to do that, we don't know right now. Um, but it's a good problem to have. It's a very exciting time in the field. Thank you so much.
Thank you for your very nice talk. So, dear colleagues, maybe somebody have a question?
Hello, I actually have a question to
Okay.
Ryan. Yeah, my name is Mikola. I'm like, from Kiev. I actually work in the field of drug discovery and actually is um, virtual screening. And I have a question about this single perturbation. So you just did all this manipulation and calculate or synthesize these molecules, or you did some maybe very expensive computational study of these compounds before the synthesis, or you did all this like transformation from carbon to nitrogen and um, just like compare the result of biochemical or biophysical screening.
Yeah,
Yeah.
it was all done, no computation. Um, the molecules were just picked. The only logic is that we would make you know a single change, and we would make all the ones that we could afford to make. The molecules were made at enamine. If we said, oh, well, can we put a fluorine here and they could make it for less than $400, then we would make it. But if it would cost us $4,000, then we wouldn't make it. But everything we could make, we made. And no thinking, just random, systematic, comprehensive perturbations. Uh, and subsequently, we have worked with Schrodinger to do free energy perturbation to see how well free energy perturbation predicts the experimental results. And that's still ongoing. We don't know the results, but at the beginning there was no computation. We just did everything we could. No
Yeah.
bias.
Thanks is actually answering my question. And like the question after you mentioned the Schrödinger, so you are trying to calculate how good FEP works based on like large data that you already have in your lab.
Correct.
As it, yeah, thank you.
Okay. Dear colleagues, other questions?
I have a question.
Yeah,
Hi,
okay.
hi, Brian. I'm an alumni of the lab. I saw this on Twitter, so I thought I'd join. First, I gotta say another amazing talk. I really enjoy listening to it. And my question is, at the end of your talk, you mentioned that you believe that soon um, docking will be kind of drowned out as the libraries continue growing by potentially false positives. And I'm wondering, how do you foresee tackling that challenge? Are you thinking of using AI or maybe docking fragments? What's your idea to continue pulling out all these potent actives?
Right. So there's two great question. It is a challenge for us. One thing that you can notice is that the what we call the cheating molecules, these artifacts, they break from the overall distribution. So their scores start to get really unusually good and very different from what you'd expect. So that's one way you can tell them. The second way is by using an orthogonal scoring function. So, this is the docking scoring function, has its problems, but you can use a second scoring function which will also have its problems, but different usually than docs. And what we have a Eugene Wu in the lab has a paper that's just come out in the Journal of Medicinal Chemistry where he tries that strategy against the artifacts that Feng Yu found, and it worked really well. We tested molecules and we were able to predict the artifacts. So I think that combination of looking at the scoring distribution and then filtering them through a second scoring function. So far seems to work. So we're cautiously optimistic there. Thanks for the question.
Sweet, thank you.
Thank you.
Hello, excuse me.
Yeah.
Uh, I'm sorry, but this question may sound a bit basic, but could you please explain how does the library itself work? Because from what I see here from your fantastic talk, the library is able to predict the position, the place where the binding occurs, and it also kind of predicts the energy. But how do we know that? Is it like from previous structural data, or is it AI, or is it some kind of semi-empirical molecular dynamics? So what is it? Where do we get that information from?
Yeah,
Thank you.
it's a physics-based scoring function. It uses so for steric fit, we use a calculation of the van der Waals energy using terms that we stole from Amber. So we use the Amber parameterization and the Amber functional form for calculating London dispersion attraction. For electrostatic complementarity, we use a point charge model of the Poisson-Boltzmann equation. So we pre-calculate all of the potential energies for the receptor are pre-calculated. So we calculate the electrostatic potential of the binding site, and then we fit, the molecule into the binding site. And, what we do is we have a potential at every lattice point in space and in the binding site, and we just multiply the partial atomic charge of the ligand by the electrostatic potential that we've calculated via Poisson-Boltzmann, and that we have something similar for calculating the cost of bringing the small molecule out of solution. We have a few other terms. They're all published, nothing secret. but they're all physical chemistry terms. So we use basically classical molecular mechanics in a static binding site or mostly static binding site and just add up the energy. So it's really classic molecular mechanics.
So you actually do optimize the position of the substrate inside like a frozen cavity, right?
We do a little bit. We do that by several ways. One is we sample a lot of different positions and a lot of different conformations. That's where the million conformations, the million placements of the molecule in the binding site come from. The second way we do it is we do once we've got a good configuration of the small molecule in the binding site, is we do rigid body relaxation of that position in the binding site, but we don't relax the receptor, and we don't, and even when we relax the small molecule's position, we've already sampled its conformations. We don't relax that any further either. So it's pretty limited relaxation.
Great, thank you. Thank you very much.
Thank you. Brian, we have a question in chat.
The chat. Okay. Let me see. Okay. Have you used AlphaFold generated structures for docking when the crystal structures of your drug target was unavailable by cryo-EM? Yes. So we just had a paper, also by the first author is J.K. Liu here at the bottom, Junken Liu, and our lab and Brian Roth's lab and Andy Cruz's lab, where we compared docking against experimental structures to AlphaFold structures when AlphaFold didn't know the experimental structure, when the experimental structures were solved after the AlphaFold database was created. And what we found there, and we tested against each target hundreds of molecules. So this was done against serotonin 2A receptor and sigma 2 receptor, which we'd worked on before and had results against the experimental structures. And then we took no refinement, took the AlphaFold structure from the database that they very kindly provided and just downloaded it, didn't do anything to it, and docked exactly as we'd done, the same library, same setup, everything the same, against the AlphaFold structures. And for sigma 2, the AlphaFold structure, by luck or not, was very similar to the crystal structure. For serotonin 2A, there were important differences, especially in the binding site. What we found was that by hit rate, AlphaFold did just as well as the crystals. We tested hundreds of molecules, so the hit rate is reliable. The hit rates were high in both cases, and they didn't differ meaningfully. And by affinity for sigma 2, we got more potent molecules from the experimental screen, but not much more potent. They bound in the same range. There's just more were low nanomolar from the crystal structure. But for serotonin 2A, by potency and efficacy and selectivity, actually AlphaFold did better. So our conclusion is that for structures where AlphaFold does well, but how are you sure? It's just as good as the experimental structures. The other thing though is that we only did this experiment on structures where we thought AlphaFold had a chance. There were other structures where the AlphaFold prediction was different enough that we just didn't think it was worth docking against. It was in the binding site, it was too different. So that's the caveat. But when AlphaFold gets it basically right in the binding site, I think it can be pretty good.
Okay. Good. So, dear colleagues, I don't see other questions. No hands, and in chat also empty. Okay, yeah.
Hi, my name is Kostin Žuravelis. Thank you for a great talk. My question is regarding single perturbation, and furthermore, so in the last slide you mentioned that the libraries will grow fast. So is it possible then in a few years when the quantum computers will be used, so we can make double, triple perturbations and so on and so forth?
The major, for sure. Yes, the major restriction is synthetic accessibility. So for instance, so we're very interested in this idea of now double perturbations, right? So we've got these molecule 278 where we just make one change. And then what you'd like to do is say, well, now let's make two changes, right? Let's put a methyl and a fluoro, or take the two places, like if I show you, yeah, sorry about this, but if we go back here, like say here, we put a methyl on and it improved things 18-fold. And here we put another methyl on and it improved things almost 300-fold. So let's try both and see if they're additive, right? Because that's another one of the great assumptions of medicinal chemistry. It's like there's three laws of medicinal chemistry. All of them are wrong, and one of them is additivity. And is it true? And so we've been wanting to do that experiment, but the problem is that the asking for the double perturbation turns out to be expensive. So we got the single changes that our friends at Enamine could make for us, and that those are the only ones we looked at. And now we're asking for the doubles where they seem interesting to us, and a lot of the time they're too expensive.
Mm-hmm.
So I think that's the major problem isn't the problem of calculation. The major problem is the problem of synthesis right now.
Mm-hmm. That's why I ask
Okay.
about quantum computers, which may possibly work more efficiently in this way.
Yeah,
Yeah.
we're better. We're bigger compute power could really help us is picking the right molecules to do these experiments on. Like we just picked the molecules we had. And I think they're probably with a larger library and a better abilities to search among them, we could pick the molecules where we could synthetically make the changes that we want to make in advance. So I think if we were doing it again from the start, we might have chosen different molecules. But we're not going to redo it. It was a very expensive experiment.
Mm-hmm. Okay, thank you.
Thank you. And we have another question in chat. Are you using only enumerated space real or Centon base to real space?
Up till now, we've only been using the enumerated space. But the the building block space, the synthon space, is very interesting to us, and we have been exploring that. And to get to the trillions, we either have to use the synthons for now. We have to use the synthon space or a machine learning version of the same thing. But we're exploring. We definitely are very interested in the synthon-based space. But all the results I've shown you are pure, just enumerated.
Thank you. Oh, dear Brian, thank you again for your very nice and fantastic talk. And I hope you will have a good day today in Frisco. So, and dear colleagues, we will meet with you on the next meeting after New Year and maybe at the end of January.
So thank you for having me. My
Thank
honor.
you. Goodbye.
Bye bye.